Hello and welcome to our water lab analysis of unknown A. So qualitative analysis is a subject interpretation of content which can be done through analytical techniques. So within organic chemistry we use analytical techniques to distinguish the functional group, so using IR, and the proton and carbon environments, so by using NMR. So water can be tested for a whole range of species by adding different solutions to form solids called precipitates. So at GCSE we'll talk about hard and soft water, where hard water contains magnesium and calcium ions. And at A level, we introduced about the solubility product and the fact silver halide ions are insoluble in water. So if we add silver nitrate to sodium chloride, we'll form a white precipitate of silver chloride, with the rest of the solution being sodium nitrate. So metal ions are separated in 5 different groups due to their ability to be identified from solution. The procedure is to separate and identify multiple ions by partitioning them out using different conditions. Group 1 ions consist of silver, lead, and mercury, with their chloride ions being insoluble in water. By adding hydrochloric acid and performing different tests on the precipitate and the centrifuge, we can identify whether silver, lead, or mercury ions are present within the water. So, how do we test for group 1 cations? So we get the water sample and add 6 molar HCl in centrifuge. Keep repeating this until no more solution precipitates out. To distinguish between lead, mercury, and silver ions, we need to separate the precipitate and the centrifugate. So, to the precipitate, we add water and heat. To test for lead ions, we take the centrifugate and add acetic acid and potassium chromate. A yellow precipitate would indicate lead is present. For mercury, we take the precipitate, then add hot H2O and 6 molar HCl. To precipitate of this, if we add 6 molar NH3 and there's a grey precipitate, we know mercury is present. Then to the centrifugate, we can either add potassium iodide or 6 molar nitric acid. When we add the potassium iodide, a creamy yellow precipitate should form, or if we add nitric acid, a white precipitate will form, indicating silver ions. Are the results. So you can see here we've got no yellow or grey precipitate, so this indicates we had no lead or mercury ions present in the solution. So on the left hand side we had a white precipitate, and this resulted from the centrifugate reacting with nitric acid. So the nitric acid was used to remove any remaining ions present in the solution, so the only ions remaining in solution were the silver ions and chloride ions from the HCl at the start. So as a result, this formed a white precipitate of silver chloride. On the right hand side we have a pale yellow precipitate, and this resulted from the final centrifugate reaction between the silver and iodide ions present within the potassium iodide. This gives a pale yellow precipitate of silver iodide. Group 2 cations are insoluble sulfides within acids. And that's because we got no precipitate initially, this highlights we got no group 2 cations within our water sample. So moving on to group 3, here are the results. So on the left hand side, we've got a red solution with a dark precipitate. This indicates that ions are present within the solution, as ions react with hydroxide ions to form iron hydroxide. Ions are also shown when we added thiocyanate ions, as this formed a dark red solution of iron cyanide. On the right hand side, we have a white precipitate in a clear solution. This indicates zinc ions are present as this reacts with the ammonia. We're expecting a red precipitate when we added acetic acid and potassium thiocyanate. However, the concentration of these must have been too small for the reaction to occur, therefore the reaction would have reacted with the ammonium chloride from the start. When we tested for group 4 and group 5 cations, which are insoluble carbonates, we got a white solution and a white precipitate. This indicates that magnesium ion is present as magnesium carbonate was formed. So as a result, from our water sample, we can conclude that we have silver, iron, zinc and magnesium ions present. As we knew we had iron ions present, we could do a spectroscopic analysis of unknown A. So we had a standard solution of Fe3 plus ions at 5.00 times 10 3 molar. 25 ml of this was diluted to 500 ml for the experiment. Therefore, the new concentration within the 500 ml is 2.50 times 10 4 molar. We took different amounts of the stock solution, then diluted them up to 50 ml, and we do this for all of these. As an example calculation, for the 4 ml, the new concentration will be 2.00 times 10 5 molar. So if we all tabulate our data, here are the results. So you can see here, as the volume of stock increases, so does the concentration of the Fe3+. and we also calculated the absorbances using the Vernier. We then did this for our unknown. So here are our absorbances and our average absorbance, and we want to find the concentration of this. So how do we do this? We have to use the relationship between the concentration and the absorbance using A equals epsilon Cl, the Beer-Lambert law, to calculate this. So looking at a graph here, we can either do this graphically or algebraically. So if you want to do this graphically, we look at our average absorbance of 0.817. We then go across the graph and down. And this gives an average concentration of 6.4 times 10 to the negative 5 to 6.6 times 10 to the negative 5 molar. Or we could do this algebraically. So if we use our equation here, we know that y is the absorbance and x is the concentration. Therefore, if we rearrange the equation to get x as the subject, we then input our numbers. Therefore, the final concentration algebraically is 6.52 times 10 to the negative 5 molar. A third method to analytical tests is something called inductively coupled plasma. So it's a method to identify the composition of a solution. It's not very good for organic molecules because they only mainly contain hydrogen, carbon, nitrogen, and oxygen. It is very good for inorganic solutions like water as it helps to identify the percentage of atoms in the water. It is very good as it can measure up to 9 orders of magnitude, and it is normally used with a mass spectrometer. As you can see in figure 8, here is an example of ICP. So as you can see here, you have your sample autoloader, you have the samples entering machine, and you have the plasma window. So as you can see here, this is the ICP calibration. So all metals were calibrated by the ICP at 25, 50, and 100 parts per million. As you can see, the only one that wasn't very good was the silver one, so this probably indicates that it will not pick up silver when analysing the water. From the ICP, we're expecting to see 4 results: the silver ions, the iron ions, the magnesium ions, and the zinc ions. So from the results, we can see we only have 2 ions. We have the iron and the zinc. The silver ions may not have shown up due to the poor calibration from the previous slide, but we should have seen a reading for the magnesium ions. When heating up the sample, the ICP goes up to a temperature of 8,000 Kelvin, which is enough energy to ionize all the metals into their ions. From the results we got, iron and zinc have ionization energies of 1,733 and 2,957 kilojoules per mole respectively to get into their stable ion state. Magnesium and silver have ionization energies of 731 and 1,450 kilojoules per mole, indicating less energy is required. So this suggests the metal was ionized past its stable state. Or the concentration of magnesium and silver was too small for the ICP to pick up, although we know they were present due to the wet water lab analysis we did earlier. From the mass concentration calculated, we need to turn this into molarity. So for iron, we know it was 91.04 milligrams per litre, so we can say that it was 0.09104 grams per litre. And we know that the MR of iron is 55.849 grams per mole. So to do this, we divide our mass concentration by the molar mass. As you can see here, the units will cancel out to give 1.63 times 10 to the negative 3 moles per litre. We then do the same for the zinc ions, so 4,802 milligrams per litre is the same as saying 4.802 grams per litre. The MR of zinc is 65.38, so divide 4.802 by 65.38, units cancel out, and we get 7.345 times 10 to the negative 2 moles per litre. In summary, qualitative analysis of water was used to distinguish the presence of silver, iron, zinc, and magnesium ions. Spectroscopic data was used to determine the concentration of iron ions in the water, and ICP was used to determine the molarity of iron and zinc from the mass concentrations, however failed to find this for magnesium and silver ions present.